So if anyone wants to talk to me afterwards, I'm more than happy to have a chat about this subject. I'm also very happy to share my slides with people if you would find it helpful to kind of read a bit more afterwards. So please feel free. My email address is there. I'll put it up again at the end. If you want to get hold of me, more than happy to provide more information. So let's start with the obvious question about what exactly is cancer. It's the most common question I get asked when people find out I do cancer research. Then people want to know what exactly is cancer. And you might think that's a bit odd because cancer is just a common disease. We all kind of know what it is, but equally we kind of don't know the details of it. And so really this is a definition. And the first thing to say about what cancer is cancer is really a collection of diseases. It's not a single disease. And we know this, of course, because we know that breast cancer is a completely different disease to kidney cancer, which is a different disease to leukemia, which is a different disease to skin cancer, and so on. Every one of those is really quite a different disease. But they share a lot of features in common. And the key feature that they all share in common is that you get an uncontrolled increase in cell number. Cells in our bodies divide and proliferate out of control. And that is the common shared defining feature of what cancer is. And a key thing I should say is that this is not about our cells proliferating really quickly necessarily, although some cancers can be aggressive and grow fast. This is really about a loss of control. It's about losing control of our own cells. And this is key because the minute life became multicellular, we want to control what our cells do in our bodies. If you're a unicellular organism, a little bacteria or an amoeba floating around in the ocean somewhere, your goal is to basically proliferate as much as possible while you've got resources and nutrients to allow you to do it. But when life became multicellular, cells joined together, and some of those cells take on specialist tasks. So in our bodies, we have a couple of hundred different types of cells that carry out different roles. Some of them are there to carry oxygen around the body, some for conducting nerve signals, some release hormones, some are more structural like bones, and some are there to move us around like our muscles. But they've got these specialized tasks. And when you have a whole group of different cells with different specialties, you need to get a measure of control over them. You need to send instructions telling these cells what to do, how to behave, and so on. So the minute we become multicellular, we have to have very strict control over the cells in our body. They can't just divide when they feel like it. They have to follow instructions and divide when they're told to divide. And we need all the cells in our bodies to be under very tight control. Cancer is what happens when we lose control. And our cells disregard signals telling them what to do and start doing whatever they feel like. And when we lose control of our own cells, they start to grow without any checks. They keep growing, we get a mass of cells that turns into a tumor, and we've got cancer. And cancer can then go to a next stage where instead of you just have this mass of cells that's growing out of control, it starts to then invade neighboring tissues and organs and even can then spread around the body. And here you can see this patient here. These glowing yellow regions are tumors which have spread to multiple parts of the body. So really the key question of cancer is, how do we lose control of our own cells? What's the process that's happening that means we don't have control over every cell in our body anymore? So to answer that question, we need to think about, well, how do we actually control our cells? What is the mechanism by which we tell cells what to do? And this looks like a complex diagram, but what it's really trying to show is that cells receive signals from their environment. And all our cells, all the time, will receive signals. It might be telling them that your blood sugar is a bit high, you should secrete some insulin. It might be saying your oxygen levels are a bit low, so you need to respond in some way. But some signals will be telling cells that you need to start to proliferate and make some new cells. That might be because the demands on your body have changed and you need more of a particular specialized cell type. It might be that you've injured yourself or damaged yourself and you need to replace those damaged cells. So signals will come into a tissue telling the cells, now is the time to proliferate and make more cells. And these particular signals that are linked to cell proliferation are what we call growth factors. They're little soluble proteins that can be secreted by other cells in your body that when that signal is received, it tells the cell now is the time to proliferate. Now, there's a particular mechanism that cells use to sense these signals and then to act upon them. And the way that cells sense these signals is they have specialized proteins embedded in their plasma membrane, so that's the outer part of the cell, called receptors. So this little U-shaped thing here with a long tail, this is a receptor. And we have many, many different types of receptors, each one of which looks for a different type of signal. In this case, this is a growth factor receptor, and it's recognized this growth factor, the signal, and as a consequence of recognizing this, we get a change in that receptor. So these signals don't cross the membrane, but the message does. What happens is the signal binds to the receptor, causes a change in the shape of the receptor that is like throwing a switch, like activating this receptor. And these receptors usually have enzyme activity. They're also enzymes, so they detect the signal, and then they can change their shape or increase their enzyme activity, which is like activating them. So we've turned this on. Now what it does, because it's stuck in the membrane, it doesn't move. So what it needs to do is activate something else that's a little bit more portable. So it binds and activates another protein. This one's a protein called GRAB2. You'll find that they all have slightly odd names. And this one now becomes activated. GRAB2's role is basically to activate another protein, in this case one called SOS, which in turn activates the next protein in the chain, and so on, until eventually we get to something that moves into the nucleus. This will activate transcription factors that move into the nucleus, turn on a whole bunch of different genes, and the cell proliferates. So this is the way we turn on cell proliferation, and then if we want to switch it back off again, all we need to do is remove that signal. If we don't have any signal, then our receptor goes back to its default state of being off. If the receptor goes off, this next one goes off, this one goes off, and so we flick our switches back to the off position. So that's how we get control. We send a signal to cells telling them what to do, activate all these switches, and then we remove the signal when we've done with it, and then all the switches go back to the off position. So that's how we control cell proliferation in our bodies. So how do we lose control? What happens in cancer is somewhere in this pathway, and it could be at any point from the receptor to something in the nucleus, one of these switches gets stuck in the on position. It gets stuck in the on position because a mutation happens, a change in the gene. Something has damaged the gene, and the protein that gets made by that gene is also altered. But it's altered in a way that means it's now permanently stuck on. So in this example here, this middle protein in the sequence, this protein called RAS, has been mutated, it's damaged, and it's stuck on. What that means is it's sending a signal to the next one in the chain, and the bottom half of this chain is now all being switched on and activated. But there's no signal present telling them to proliferate. And if we add a signal, we'll activate these ones and drive proliferation. But if we remove the signal, we switch these off, but this one stays on. And so the consequence of this switch being stuck on is the cell is constantly being driven to proliferate. It thinks it's receiving a signal when it isn't. And most importantly, we've now lost control, because whether the signal is there or not, the cell's still proliferating. It's not listening to its instructions, it's just undergoing constant proliferation. And this is the beginnings of cancer. We've now lost control of this cell, and it's constantly proliferating, and we're getting an increasing mass of these cells. Now this is what we call an oncogene. That gene that got mutated, this one in the middle here in this example called RAS, that's what we call an oncogene because it's a gene linked to cancer. And any of those proteins in that pathway is a potential oncogene. A mutation in the gene that makes those proteins, if it was the receptor or any of the other ones, could do the same job. It would be stuck in the on position, and it would be driving more and more cells to proliferate. So here we've got an example where we've got a normal copies of our genes. One of them becomes mutated. It gets stuck in the on position, and it's like hitting your foot on the accelerator, and you can't take it off again. It's stuck in that position, driving faster and faster cell growth. And the example I give is not just a random example. That protein I mentioned called RAS, right in the middle of that pathway, that's the most commonly mutated oncogene in human cancers. Around about 20 to 30% of all human cancers, irrespective of where they start, have a mutation in that protein in the middle, this one here called RAS. So it gets stuck in the on position, and we see constant proliferation and growth of our cells. That is the beginnings of cancer. So really, we now have to think about, well, what's causing that? Why is this protein getting stuck in the on position? Why has it got mutated and now we've lost control of the cells? Well, the short answer to that is our DNA is being damaged constantly. Our DNA is always being damaged in some way. Every single cell in your body is being damaged about 10,000 times per day, every single day of our entire lives. Masses and masses of DNA damage. Luckily, we have a whole variety of DNA repair mechanisms, specialized enzymes that can repair that damage. And these repair mechanisms are amazing. They repair it with incredibly high fidelity, almost perfectly. But the key word in that sentence is the word almost. The repair mechanisms are not perfect, and every now and then it misses a piece of damage. It doesn't get repaired properly. And when that happens, we've got what we call a mutation. We have a permanent alteration in our genome. A gene has been damaged, not repaired properly, and every time that cell divides, that mutated gene will be copied and passed on to its daughter cells. So we get lots of damage, we don't repair it all, and the stuff we don't repair now becomes a mutation that we're stuck with. One of the key things to remember about mutations, though, is that they occur randomly. They're not directed. We can have a mutation anywhere in our genome. Now, we have about 20,000 genes, just over 20,000 genes in our genome, and we also have a lot of DNA that doesn't code for anything, doesn't make proteins. Some people call it junk DNA. So we've got a lot of DNA, and a mutation could occur randomly in a region of DNA that doesn't do anything, or isn't important, or is in a gene that's not linked into cell proliferation. So it isn't just a case of any random mutation can give you cancer. It's often very specific mutations that you need. You often need to have a mutation in just one of those genes linked to cell proliferation that I showed, and it has to be a mutation that actually causes it to be stuck in the on position. A lot of mutations won't do that. A lot of mutations might turn it off, in which case you won't get cancer. A lot of mutations may have no effect, in which case you won't get cancer. It has to be a mutation in one of those key genes, in just the right part of that gene, to cause that switch to be stuck on. So mutations are key, but it's a probability that's linked to cancer. It's not you get a mutation, you'll get cancer. It has to be in just the right part and the right type of mutation for it to link with cancer. So because it's now about luck, odds of where those mutations hit, we need to think about what causes those mutations, because anything that increases the rates of mutation will increase our risk of developing cancer. And there are lots of things that can cause DNA damage. We can expose ourselves to radiation. Radiation can break DNA strands, and the most common radiation that we'll experience will be ultraviolet radiation from the sun. So going out and sunbathing too much, we're exposing ourselves to UV radiation that can damage the DNA, principally in our skin cells. We can be exposed to viruses, and lots of viruses, particularly retroviruses, insert their genetic material into our own. They insert the viral genome into our own DNA, and when they do that, they can cause damage, and mutations come along with that. And then there are a whole variety of chemicals that we can expose to in our diet, in our lifestyle. Smoking, for example, when we smoke, we inhale lots and lots of chemicals that can damage our DNA. Or we can be exposed to things in the workplace, and a good example of that would be things like asbestos, things that you inhale that can damage DNA and damage cells. To a certain extent, we can mitigate against some of these. We can try to reduce our exposure, stay out of the sun, use sun creams, avoid contact with some of these viruses or have vaccines against them, and reduce our exposure to known cancer-causing chemicals. But there's two on the list that we really can't do anything about. And this is really due to the fact that there are no biological processes which are flawless and perfect. Every single biological process has the risk of errors. And not only can you damage DNA from all of these types of radiations and chemicals, but DNA can be damaged just when we copy it. Sometimes mistakes creep in. DNA is our chromosomes are long strands of DNA. it's easy to make a mistake when you copy the DNA to make new cells. When you repair the DNA, sometimes you make mistakes when you repair it. So that can just happen naturally. We'll get mutations just from the process of making new cells. But also we can get mutations just from being alive. As our cells go through metabolism and make energy from the food that we eat in the mitochondria, we break down our food, we use it to generate energy for life to happen, but the process of making that energy occasionally makes, as a side product, molecules called free radicals. This is an oxygen molecule with a missing electron that's a highly reactive molecule, and these reactive molecules can bounce into DNA strands and break them and cause damage. So there's not a lot we can do about that. If we're alive, we're going to be generating these things that cause DNA problems. And so really this is now, again, about probabilities. We are always damaging our DNA. We're not repairing it perfectly. But anything that adds to that, anything that we expose ourselves to that increases the rates of mutation will increase our chance of getting cancer. So if that was basically the entirety of the story, then actually, A, we probably would have cured cancer a long time ago, but B, we'd all have a problem of developing cancer at a very young age. Because if it was just about you acquire mutations until by sheer bad luck, one of them happens in one of those key genes, and then the switches are on permanently and we get cancer. If that was all it was, then we will be riddled with cancer before we could walk. We actually don't get cancer until much later in life as a whole, and the reason for that is we have a whole range of anti-cancer defenses that protect us when our cells start to go a bit out of control. So even when they've got these mutations and they're dividing out of control, we have ways in our body of stopping that from happening. And one of the key ones is these things called telomeres. Now, telomeres are little regions of DNA at the ends of our chromosomes. You can see them here in yellow in this diagram, and these are actually people's chromosomes. You can see the little yellow blobs at the ends of the chromosomes. They're like little caps on the ends of chromosomes, and they protect those chromosomes. But the key thing that happens is every time a cell divides, that little cap gets a bit smaller. Little chunks of DNA are carved off the end every single time a cell divides, until eventually we lose that cap completely. And what that means when you get to that stage is the enzymes that copy DNA can't bind anymore. They can't bind to the DNA. They need actually to bind to this cap to then start copying the DNA. So when this cap gets too small, that doesn't happen, and it's literally physically impossible to make any more DNA, and therefore you cannot get any more cell division happening. The cell cannot divide because it can't make any more DNA. So this is actually really primarily an anti-cancer defense mechanism. It's a way of stopping cancer in its tracks. If you've got a cell that has that mutation in that oncogene, like RAS, after it starts proliferating, it's going to get to a point where all the telomeres are too short and no cells can divide anymore, and it stops. And the limit for this is usually somewhere between 40 to 60 cell divisions. So that's not very many. When the cell goes out of control, 40 to 60 times of dividing later, when it's probably still less than a millimeter in size, it hits this roadblock. Cannot copy any more DNA, cannot continue to divide. So this is a way of preventing cancer. There's also potentially a reason why we age, because some of our cells just lose the ability to continue to proliferate. But that's kind of a side product of really trying to prevent cancer from developing. Now, of course, because cancer does eventually develop, you know that there must be a way around this that cancer exploits. And there is, because there are cells in our body called stem cells, and those stem cells are effectively immortal cells. They can live with us our entire lives, and they can divide indefinitely. They do not have this same limit. And the reason they don't have this same limit is they have a special enzyme called telomerase that rebuilds the telomeres every time they divide. So in a stem cell, this never gets shorter, it always stays the same length, and they have no limit on the number of cell divisions. Now that telomerase enzyme is completely silenced in all cells apart from stem cells, but it gets reactivated in cancer. So you need another mutation in the cancer cells that then turns this gene back on, and then the cells can start to proliferate again. But that means we need a second mutation. We needed a mutation in one of those oncogenes, in a very specific part of an oncogene, to start the process rolling. Now we need another very specific mutation to turn on telomerase to allow us to continue to divide. And that is a very common thing that happens in cancer. Here's a couple of cancer cells down in the microscope, and these are a very famous cancer cell line called HeLa cells. One of the first human cell lines that were ever cultured, taken from a patient with cervical cancer called Henrietta Lacks, hence the HeLa. She died in the 1950s from this cancer, but her cells are still growing. Those cancer cells are immortal because they've reactivated this telomerase enzyme. They have no limit on their cell proliferation, and they will continue to keep going for hundreds or thousands of years without stopping. That's what we're facing with cancer, uncontrolled cell proliferation without limit. Luckily, we have even more anti-cancer defenses though. So at this point in the cancer, they've had to have 2 different mutations in very specific genes. By this point, by sheer chance, you've probably had hundreds or even thousands of mutations in other irrelevant genes or non-coding parts of the genome. So our genome is starting to get pretty badly damaged. And at this point, we have what we call tumour suppressor genes that sense all this DNA damage, and their function is then to stop proliferation. They can physically shift the cell back out of mitosis, put it into what we call a quiescent state where it doesn't divide, or they can cause the cell to self-destruct. They activate a self-destruct mechanism called apoptosis that destroys the cell, and as a consequence of which the cell is dead and all the mutated DNA is dead with it. So there are a whole variety of these genes which are constantly scanning the genome, looking for DNA damage, and when they find it, they can slam on the brakes and potentially even self-destruct. Now this is good news because for cancer to get around this problem, you can't just have a mutation in one of these genes, because for every gene in our cells, we have 2 copies of every gene. So switching off one copy isn't enough. It is in the oncogene. In the oncogene, mutating one copy so it's stuck on is enough to drive proliferation. But wiping out one copy of a tumor suppressor gene leaves another one capable of initiating the braking system. So you actually need to have 2 mutations in the same gene at the same time to overcome this anti-cancer defense mechanism. And the most common one of these that gets mutated is a protein called p53, and here it is bound up to a sequence of DNA. So DNA is in blue and green, and the p53 protein is binding to DNA, looking for signs of DNA damage, and if it gets mutated, it can no longer do that. So now for cancer to have developed, we need a mutated oncogene, a mutation that turns on telomerase, and 2 mutations that switch off this tumor suppressor gene. There are also many other things that we have that protect us from cancer, which I don't have time to talk about, but the general principle is we have many things that protect us. And one last thing I wanted to mention that also protects us is the fact that our cells are normally stuck in a particular part of our bodies. They don't move around randomly, with a few exceptions like our immune cells. Our cells tend to stay put. They're connected to other cells and surrounded by a kind of scaffolding system called the extracellular matrix. And that tends to hold cells in place. Now, clinically, the biggest problem with cancer is when it spreads around the body. If it stays localized and doesn't spread, it can be relatively simple to treat. You can just surgically remove the tumor. And if you remove all the cancer cells, you've cured that person of cancer. If it's all in one place and it's not easily accessible by surgery, you can use radiotherapy to target it instead, or beam of radiation in the tissue. But once it spreads around the body, we have very few treatment options. It's very difficult to treat the cancer. So for that to happen, though, the cancer cells have to acquire a whole range of new capabilities. They have to learn to break away from the tumor, start moving. They have to move through this scaffolding system, often by breaking it down and tunneling their way through it. They need new enzymes to be activated. They then get into the circulation. As you can see here, these are cancer cells in the bloodstream. And the bloodstream is a very hostile environment for cancer cells because they're not well adapted to it. A lot of shear forces and high pressure kills most cells that get into the bloodstream. So they have to learn to survive. And then they get out in their distant site. Here you can see in little yellow, this is liver cancer cells that have found their way into somebody's brain and are now growing in the brain. But that is quite a difficult thing for them to do because they're adapted to the liver. They're expecting to see other liver cells and nutrients specific to liver cells, and when they get to the brain, they don't find them. So for this to have happened, we need a whole variety of additional mutations. So really, for cancer to become a problem, what we're seeing is an oncogene mutation, telomerase mutation, 2 tumour suppressor gene mutations. They have to mutate to be able to spread away from the first tumour, learn to survive in the circulation, and then learn to survive at the distant site. That requires a lot of additional mutations. And that is why cancer is primarily a disease of older age. It takes a long time for cancer to acquire enough mutations to overcome all of our anti-cancer defenses and become a clinical problem. And so normally for most people that wouldn't be something you'd encounter until you've been alive for 50, 60, 70 or more years of life. That's how good our anti-cancer defenses are. And indeed, we're now able to sequence the genomes of tumors, and we can look at how many mutations are there in cancer. And this is one of the very first studies. This is a study that was looking at lung cancer, and what they found is that there are 23,000 mutations in the lung cancer tumor cells compared to the normal cells in the lung. 23,000 mutations. That is how many mutations it took to overcome our natural anti-cancer defenses. Huge numbers of mutations. We could tolerate all sorts of DNA damage before cancer becomes a problem. Most other tumors we've now sequenced run into the thousands as well. So we are actually, as a species, pretty cancer resistant. It takes a lot to generate cancer. It's just that we're living longer lives now, and so we're getting to a point where those mutations do accumulate and cause us more and more problems. Now, in clinical terms, we've been getting a lot better at treating cancer. All of the stuff I've just talked about is things that we've learned about cancer biology. We understand now the genes which are mutated. We understand the processes that happen in cancer, and that's allowing us to get better treatment options. If we look at the survival times of cancers, a typical patient diagnosed with any random cancer in the 1970s would have on average survived about a year after diagnosis. By the time we got to the early 2000s, an average cancer patient would survive nearly 6 years after diagnosis. And this lifespan after diagnosis is increasing all the time. We're just getting better and better at treating cancers. We don't have a cure, but we are getting better at allowing patients to survive with cancer for longer and longer. That, though, is not true of all cancers. I'll draw your attention to this graph at the bottom right. This dotted line here is basically the same graph here. It's showing the survival time of all cancers when we look at them as an average. Anything below this line, like these ones down here, are doing much worse than average. And you notice some of them, this is pancreatic cancer here, this is brain cancers, we've got lung cancer on the list. It's almost a flat line over the last 40 odd years. Very little improvements in those cancers. We're just struggling with those cancers in particular, even though we're making good progress with other cancers. And so we're really looking now to try and develop better treatments. And I appreciate I'm running a little bit short of time, so just very quickly, the kind of treatments that we'd be looking at, frontline treatment, it'd always be surgery. If the cancer hasn't spread and it's in a place that you can operate on, then surgery has got your highest chance of curing the patient and allowing a normal life after that. If you cannot operate on it, so for example, a lot of lung cancers are very difficult to operate on, so are some brain cancers, you might use radiotherapy where you can target a beam of radiation deeper into the body to try and damage the cancer cells. And the aim here is that the radiation would destroy the DNA, make it impossible for the cell to proliferate, and may also activate these self-destruct mechanisms so the cells will kill themselves. If those are not possible, if the cancer is spread, then we fall back onto chemotherapy. And chemotherapy is a series of drugs that we can give. You inject them into the bloodstream. They circulate through the body, so they should be able to target cancer cells wherever they are. The problem is we really don't know how to identify a cancer cell compared to a normal cell. They are cells. They're us. They're very similar to the normal cells. So the way that chemotherapy drugs generally work is by targeting proliferating cells. Anything that's actively proliferating is more sensitive to chemotherapy drugs. And hopefully that would be mostly the tumour cells, but it also is cells in our body which are naturally proliferating a lot, like our hair follicles, the lining of the gastrointestinal tract and so on. And so you get these common side effects like hair loss and nausea and vomiting. And eventually the big thing that stops you from giving chemotherapy is bone marrow toxicity and effects on a patient's immune system. But our underlying knowledge of cancer biology and what's going on in cancer is now leading to new drugs which are being developed that can switch some of these mutations back off. Those oncogenes stuck in the on position, we're now learning to be able to switch them back off. We're now learning to get new drugs that can target things like the immune system, the metabolism of cancer, and all sorts of new options for treating cancer. So I fully expect that that survival curve will continue to improve over the next few decades. So this is just a whole bunch of information I normally give at the end of a lecture. And just to reiterate that if anybody wants a copy of my slides, you're more than welcome to just send me an email, and I'm more than happy to chat about it if you're here for the rest of the day. But apart from that, thank you all very much for your attention. Thank you.